Human Genome Project. The Human Genome Project is a global, long-term research effort to identify the estimated 30,000 genes in human DNA, deoxyribonucleic acid, and to figure out the sequences of the chemical bases that make up human DNA. Findings are being collected in databases that researchers share. In addition to its scientific objectives, the project also aims to address ethical, legal, and social issues, which the project refers to as ELSI. The project will make use also of results from the genetic research done on other animals, such as the fruit fly and the laboratory mouse. Research findings are expected to provide a dramatically greater understanding of how life works and specifically how we might better diagnose and treat human disorders. Besides giving us insight into human DNA, findings about non-human DNA may offer new ways to control our environment. A genome is the sum of all the DNA in an organism. The DNA includes genes, each of which carries some information for making certain proteins, which in turn determine physical appearance, certain behavioral characteristics, how well the organism combats specific diseases, and other characteristics. There are 4 chemical bases in a genome. These bases are abbreviated as A, T, C, and G. The particular order of these chemical bases, as they are repeated millions and even billions of times, is what makes species different and each organism unique. The human genome has 3 billion pairs of bases. Studies of this project will be undertaken under 3 steps. 1. Mapping. 2. Sequencing. 3. Functional analysis. Mapping of human genetics is difficult due to the following reasons. 1. Ethical reasons. 2. Length of breeding cycle is only 15 to 16 years. 3. Inability to carry on controlled breeding. Few interesting features of human genome are: there are more than 3.2 billion base pairs. Human genome is estimated to have 20,000 to 25,000 genes and to determine the sequences of 3 billion chemical base pairs that make up human DNA. Within the human population are millions of single base differences called single nucleotide polymorphisms, SNPs. The human genome bears gene-rich areas separated by gene-poor areas called gene deserts. About 45% of our genome is made up of transposons. Only 2% of genome codes the proteins. To improve tools for data analysis. Use of concerned technology to sectors like industries. Address the ethical, legal and social issues, ELSI, which may appear due to HGP. The human genome contains 3,164.7 million nucleotide bases. Largest known human gene is dystrophin, which bears 2.4 million bases, with average gene having about 3,000 bases. Function of 50% of discovered genes are yet to be found.